Ireland's first ancient genomes were meant to prove a story everyone already believed. Instead, they proved almost the opposite. Popular history credits Ireland's ancestry to an Iron Age Celtic invasion, sweeping in from Central Europe, arriving somewhere around 500 BCE, with Latin art, warrior culture, and the Celtic languages that still echo in Irish place names today. It's the version taught in schools, repeated in tourist brochures, printed on souvenir maps. It is also, according to the DNA itself, not what happened. Cassidy and colleagues sequenced Ireland's first ancient genomes back in 2016, and the results didn't show a mass Iron Age migration event stamped into the population's genetic record. There's no clean signature of a Celtic wave sweeping across the island and replacing what came before it. What the genome actually shows is older and stranger. The insular Atlantic genome, the shared genetic profile running through modern Irish, Scottish, and Welsh populations, traces back to the Bronze Age instead, roughly 4,000 years ago, over 1,000 years before anyone identifiable as Celtic would have set foot on the island. Layered onto that timeline is a number that should stop anyone reading it. Modern Irish Y-chromosome studies put the frequency of a single lineage, R1b-M269, at roughly 81.5%. One 191 sample study measured it even higher, at 85%. That is not what a layered history of repeated invasions looks like. A truly layered past leaves a mosaic, different lineages stacked unevenly across centuries of contact and conquest. What Ireland shows instead is one haplogroup dominating almost the entire population. A genetic signature so uniform it looks less like accumulation and more like replacement. The Celtic label hasn't disappeared. It survives, comfortably, as a linguistic and cultural term, describing language, art, and identity. But geneticists and historians studying this same question have openly called it a decades-long, unresolved controversy. Whether the transformation of Europe's Atlantic edge was driven by real migration, actual people moving in and taking over, or simply cultural adoption, ideas and language spreading without a single new person ever crossing the Irish Sea. That controversy didn't start with genome sequencers. It started decades earlier with something far cruder and far more overlooked: blood. Before anyone could sequence a genome, before anyone could plot a single Y chromosome on a graph, the first hint that Ireland wasn't genetically uniform came from something as simple as blood typing. Researchers running ABO and rhesus blood group surveys across the island stumbled onto a pattern nobody was specifically looking for. These early serology studies revealed a general east-to-west cline running across Ireland, a gradual shift in blood group frequencies from one coast to the other. It wasn't dramatic. It wasn't even fully understood at the time, but it was there, decades before anyone had the tools to explain why. It took decades more before that crude cline could be checked against anything more precise. When dense genome-wide SNP data finally arrived, it confirmed something remarkable. The blood typing pattern from decades earlier mirrored the far more detailed Y chromosome results that came later. The old serology wasn't noise. It was a low-resolution version of a signal that turned out to be real. Genome-wide analysis added another layer to the picture. Irish haplotype diversity came back measurably reduced compared to other European populations. Both runs of homozygosity and linkage disequilibrium showed up elevated, hallmark signs of a population that had, for long stretches, stayed relatively closed off from outside gene flow rather than continuously mixing with its neighbors. What the early researchers couldn't do was place any of this on a map. Blood group surveys weren't geocoded. Scientists could detect that Ireland was internally different, that the island wasn't one uniform genetic block, but they had no way of showing where those differences actually lived. Which counties, which coastlines, which regions carried which signal. That gap stayed open for decades. It didn't close until 2017, when the Irish DNA Atlas project finally combined generations of genealogical record-keeping with modern genome sequencing, giving researchers, for the first time, a way to place Ireland's genetic structure precisely onto its own geography. The blood group data itself was crude, unrefined by any modern standard. But it was the first hard evidence, gathered long before anyone could sequence a single strand of ancient DNA, that Irish was never one genetically uniform population to begin with. And that same blood typing era is exactly where the Basque comparison first took root, decades before any Y chromosome study would come along and confirm it was real. I need to keep the boundaries strict per segment. Let me now write segment 4 and 5. Basque populations carry ABO type O frequencies above 50%, a striking concentration by any standard, while type B shows up as extremely rare across the same population. The highest O-type numbers weren't scattered evenly across the Basque country either. They clustered hardest in the most remote areas, places like Seoul, where the Basque language itself has survived in its most unbroken form, untouched by the linguistic pressures that reshaped the rest of Western Europe. Early 20th century researchers noticed something else entirely. Decades before anyone could sequence a single genome, the Irish, the Scots, the Corsicans, and the Cretans all carried unusually high O-type frequencies of their own. 4 populations scattered across opposite corners of the continent, all sharing the same blood type pattern. That overlap was enough to trigger speculation about some shared ancient kinship connecting these groups, long before anyone had a single tool capable of actually testing whether the idea held up. The timeline here matters. The rhesus factor, the marker most people now associate with the Basque Atlantic story, wasn't even discovered until 1937. The theory of a Basque-Atlantic connection predates it entirely, built instead on ABO blood typing alone. Northwest Ireland carries its own striking echo of the Basque pattern, independent of blood type altogether. Regional samples there have recorded R1b, the same Y-chromosome marker dominant across the Basque country, exceeding 98% in some local populations. 2 separate lines of evidence, discovered independently, decades apart, using entirely different methods, both point toward the same geographic outlier. In population genetics, that kind of independent corroboration is unusually strong. That's precisely where the science and the folklore start to pull apart. One of these threads is peer-reviewed, built on published frequency data and dated haplogroups. The other belongs almost entirely to internet mythology. One published hypothesis takes the Rh-negative pattern somewhere completely different. It proposes that Rh-negative frequency correlates with the regional prevalence of Toxoplasma gondii, a common intracellular parasite found across much of the world. The theory suggests carriers of the Rh-negative allele may carry a selective advantage tied to greater parasite resistance, one strong enough to offset the well-documented reproductive risk that comes with Rh-incompatible pregnancies. If that holds, it would explain something that otherwise looks strange. Rh-negative frequency shows up at elevated rates in populations with no demonstrated common ancestry at all, Basques and certain Jewish populations among them. Under this model, Rh-negative frequency isn't necessarily the fossil trace of one ancient bloodline spreading from region to region across millennia. It can arise independently, wherever the same selective pressure happens to exist, in populations that never had any contact with one another. That directly complicates the simpler reading of the Basque-Irish Rh-negative overlap, the one that treats it as proof of ancient shared lineage. It doesn't erase the Y-chromosome data, which still supports a real historical connection between these 2 populations. But it separates 2 questions that keep getting collapsed into one. Population geneticists treat these as genuinely distinct, testable problems. Is the lineage itself shared, a Y-chromosome question, answerable through haplogroup dating and mutation age clustering? And separately, is the elevated trait frequency shared for the same underlying reason, a selection pressure question, answerable through parasite prevalence and reproductive risk modeling? 2 different methods, 2 different kinds of evidence, 2 different answers. Conflating those 2 questions is exactly how a real, peer-reviewed genetic finding gets flattened into something far simpler and far less accurate. A nuanced, 2-part scientific picture collapses into a single tidy claim. The kind that spreads easily, precisely because it no longer resembles the actual research it came from. And much less accurate, undersells what certain corners of the internet have actually done with this same data. Because Basques carry both the highest Rh-negative frequency on record and a documented language isolate, they've become the anchor point for a long-running set of fringe theories about human origins. One narrative ties Rh-negative blood directly to Cro-Magnon ancestry from the Upper Paleolithic, treating it as physical evidence of some deep, untouched lineage stretching back tens of thousands of years. A more extreme version pushes further still, claiming Rh-negative carriers descend from a so-called Hyperborean race, a mythologized, non-scientific origin story with no basis anywhere in the peer-reviewed literature. These narratives typically reach beyond the Basque country too, pointing to the Atlas Mountains of Morocco and the Canary Islands as other supposed Cro-Magnon holdouts, based purely on similarly elevated Rh-negative rates in those regions. None of this appears in the actual genetics literature. The real papers describe allele frequencies, selection pressures, and precisely dated haplogroup mutations. They do not describe lost human subspecies or hidden bloodlines waiting to be uncovered. The gap between the real 2021 Basque Ireland Y-chromosome study and this folklore is the same gap that sits between a measured MDS plot and a story built specifically to feel mysterious. It's worth separating these cleanly because the real finding doesn't need the mythology attached to it. A documented language isolate, an independently dated Y-chromosome lineage, a measurable frequency gradient across Iberia, these are strange enough on their own terms, without borrowing language from pseudoscience to make them feel bigger than they are. The actual data holds its own weight. What the real data needed next wasn't more theorizing. It needed 4 actual skeletons, pulled directly out of the ground in Northern Ireland. In 2016, Cassidy and colleagues at Trinity College Dublin published the first ever ancient whole genomes recovered from Ireland, appearing in PNAS. The oldest sample belonged to a Neolithic woman from Ballynahatty, County Down, dated to between 3343 and 3020 BC, sequenced to 10.3x coverage, a remarkably high-resolution result for an ancient genome of that age. Her genetic profile placed her firmly alongside other Middle Neolithic farmer samples recovered from Germany, Spain, and Scandinavia. Her ancestry traced to Near Eastern-derived farming populations, not to anyone native to Ireland itself. Reconstructions based on her genome suggest she had black hair and brown eyes, a physical profile strikingly different from the red-haired Celtic stereotype now associated with Ireland in the popular imagination. Her presence confirms something specific about how farming reached Ireland in the first place. It arrived through migration, real people moving from continental farming populations onto the island, not through local hunter-gatherers independently adopting farming techniques on their own. This detail carries particular weight for the Basque comparison. Iberian farming populations and Irish Neolithic farming populations both descend from the same broad westward agricultural expansion that began in Anatolia. That shared farming migration origin predates the Bronze Age R1b expansion by thousands of years, forming a second, entirely independent layer of ancestry connecting these two regions at opposite edges of the Atlantic. Then, 1,000 years later, 3 men buried on Rathlin Island completely overwrote what she represented. The same 2016 study sequenced 3 early Bronze Age men recovered from a cyst burial on Rathlin Island, dated to between 2026 and 1534 BC. Genetically, they were almost unrecognizable next to Ballynahatty, carrying a distinct Caucasus ancestry component tied to Pontic-Caspian steppe populations, a signature entirely absent from the earlier Neolithic sample. One of the Rathlin men carried the C282Y hemochromatosis mutation, the same gene variant responsible for Ireland's modern status as the country with the world's highest frequency of the disease, sometimes nicknamed the Celtic disease as a direct result. The Rathlin men also carried blue eye alleles, and the most common modern iris Y-chromosome type, physical and genetic traits still dominant across Ireland today, 4,000 years later. Lead researcher Laura Cassidy described the genetic affinity between these Bronze Age genomes and modern Irish, Scottish, and Welsh populations as the strongest of any comparison in the study, dating the insular Celtic genome to roughly 4,000 years ago. Statistical d-tests confirmed that Ballynahatty and other Middle Neolithic samples form a genetic clade that excludes the Rathlin men entirely, evidence that this wasn't gradual drift accumulating over centuries. It was a measurable population replacement. Ireland's Neolithic and Bronze Age populations turn out to be, genetically, close to 2 different peoples occupying the same island 1,000 years apart. That replacement didn't happen in isolation. It was part of a wave that hit the entire British Isles at once. The scene shifts north, across the water, to Britain itself. In 2018, Olalde and colleagues published genome-wide data from 400 individuals in the journal Nature, tracing what researchers now call the Beaker phenomenon. Beaker culture-associated migrants, carrying steppe-derived ancestry, arrived in Britain roughly 4,400 years ago, bringing with them a distinctive style of pottery and a genetic signature that hadn't existed on the island before. Within a few hundred years, this migration was associated with the replacement of approximately 90% of Britain's existing gene pool. That number alone reframes the entire story. This wasn't a gradual blending of populations over centuries. It was something closer to wholesale turnover. R1b M269, the same Y-chromosome lineage now dominant across Ireland, Britain, and the Basque Country, was essentially absent in Neolithic Britain before this event took place. After the Beaker migration swept through, more than 90% of male individuals in Britain, and in the populations that followed, carried R1b. Iberia tells a completely different version of the same story. Iberian Beaker culture sites show the opposite pattern entirely. Local, pre-existing Y-chromosome lineages persisted there, largely undisturbed. The pottery style spread across Iberia, the artifacts look the same, the cultural markers match, but the people underneath stayed put. In Iberia, Beaker culture spread through cultural exchange. In Britain, it spread through actual migration, real people arriving and displacing what came before them. Geneticist David Reich, co-senior author on the study, called it one of the clearest ancient DNA cases of a phenomenon researchers had long suspected but rarely could prove so cleanly. The same object, the same pottery style, spreading through two entirely different mechanisms depending on where you look. Pots, in other words, do not always travel hand in hand with people. Sometimes an idea moves, sometimes a population moves, and distinguishing between the two requires exactly the kind of genome-wide data this study provided. One lineage did the traveling in both directions, into Britain and into Iberia alike, and it started its journey a lot further east than either Ireland or the Basque Country. The M269 mutation that defines R1b traces back to the Near East, moving alongside early farming populations as they spread toward Anatolia and eventually out onto the Pontic-Caspian steppe. From there, a downstream mutation, S116, also known as P312, is estimated to have arisen roughly 5,000 years ago, most likely somewhere in what is now France. From France, this lineage split in two directions. One branch fed into the population that would eventually become dominant across the Basque Country and Iberia. The other branch fed into the population that would seed Britain and Ireland through the later Beaker migration. Two branches, one shared route, diverging somewhere in prehistoric France before heading toward entirely different corners of Atlantic Europe. In Iberia specifically, R1b shows up mainly as RS116, reaching an average of 80% across the Basque provinces. A derivative lineage within that branch, DF27, believed to have originated inside Iberia itself, peaks at 63% in the Basque Country before dropping to 40% in neighboring Galicia. That's a real, measured gradient across the landscape, not a flat, uniform block of ancestry stretching evenly across the peninsula. This detail sharpens the entire Basque story considerably. The Basque genetic isolate framing is more specific than it sounds on the surface. Basques share the broad R1b route with Ireland, the same deep ancestral trunk, but they carry a regionally distinct sub-branch, DF27, that developed after the split occurred. Shared origin, separate development. The timing matters just as much as the geography. That shared route split roughly 5,000 years ago, close enough to the Bronze Age turnover recorded on Rathlin Island, that both events plausibly belong to the same broader migration era sweeping across Atlantic Europe. That shared route is exactly what the 2021 Basque Y-chromosome team set out to measure with real precision. The 2021 scientific report study didn't treat Basque as a single genetic block. Researchers sampled 3 provinces separately, Álava, Gipuzkoa, and Vizcaya, each analyzed as its own distinct population. RS116 frequency measured 75.0% in Álava, 86.7% in Gipuzkoa, and 87.3% in Vizcaya, genuine internal variation within a population that popular narratives usually flatten into one single statistic. Local age estimates for the RS116 mutation differed by province too. In Álava, researchers calculated 3,975 years, plus or minus 303. In Gipuzkoa, 3,680 years, plus or minus 345. In Vizcaya, 4,553 years, plus or minus 285. 3 separate provinces, 3 independently dated origins for the same lineage, all landing within roughly the same broad window of time. Pairwise RST genetic distance calculations added another layer. They showed close Y-chromosome affinities, not only among the 3 Basque provinces themselves, but between all 3 provinces and the male populations of Ireland and Gascony. In the study's multidimensional scaling plot, Ireland's population segregated directly within the Basque cluster, specifically closest to Gipuzkoa, closer to Gipuzkoa than Gipuzkoa sat to its own neighboring provinces of Álava and Vizcaya. Gascony, in southwest France, clustered near this same group as well. The finding isn't quite the story of one single isolated region on Earth carrying a unique signature. It's a remarkably tight three-way cluster, spanning 2 countries and a full sea crossing between them. The researchers frame this pattern as consistent with a Bronze Age population replacement event connecting these regions directly, rather than something explainable as pure coincidence or ancient shared ancestry, simply diluted thin over millennia. For decades, geneticists already had a name for this pattern. They just assumed it meant something completely different. Before this newer data existed, researchers including Hill, Wilson, and McAvoy had already identified a shared Y-chromosome pattern connecting Basque populations with the Western Celtic populations of the British Isles. They named it the Atlantic modal haplotype. The AMH is defined by specific SNP and microsatellite markers within R1b, and it stands as the most frequently occurring haplotype among males across Atlantic Europe. For years, this shared pattern carried one dominant interpretation. Researchers read it as a Paleolithic legacy, evidence of a population that had survived largely undisturbed at the western edge of the continent since the last ice age. A genetic time capsule stretching back tens of thousands of years. One older analysis went so far as to call Basques nearly identical, in Y-chromosome terms, to predominantly Celtic populations like the Irish and Welsh, without yet knowing the mutation's actual age. The Olalde 2018 Beaker data undercut that Paleolithic refuge interpretation directly. If R1b entered Britain through a Bronze Age replacement event roughly 4,400 years ago, the shared haplotype simply cannot be a 15,000-year-old survival story. The math no longer works. The ancient refugium framing wasn't wrong about the core observation that Basques and Irish share a deep genetic affinity. It was wrong about when that affinity was established, and wrong about how it got there. Better dating technology overturned a decades-old theory without discarding the underlying observation that started it in the first place. Move south along the Atlantic coast, and the same pattern keeps showing up, region after region. Galicia, tucked into the northwest corner of Spain, carries R1b DF27 at roughly 40%, lower than the Basque Country's 63%, but still a substantial regional presence of the same lineage. Genetic distance analysis in the 2021 study placed Gascony, in southwest France, among the populations most closely related to both the Basque provinces and Ireland, a second French connection reinforcing the first. Neither Galicia nor Gascony shares the Basque language isolate. Both regions speak languages with clear Indo-European roots, nothing like Euskara. The genetic overlap connecting these regions to Basque and Irish populations exists entirely independently of any linguistic connection, a signal running beneath language rather than alongside it. This regional spread suggests the Bronze Age R1b expansion moved broadly along the Atlantic coastline, rather than settling into one isolated Basque pocket and staying there. Coastal seafaring geography likely played a real role in that spread. These are all regions connected historically by maritime trade and travel routes running along the Atlantic facade of Europe, a natural corridor for people and lineages to move through generation after generation. Modern DF27 distribution essentially maps out as a gradient across that same coastline. Highest in the Basque Country, declining steadily through Galicia, with related lineages continuing north into Brittany and on into the British Isles. None of these regions, Galicia or Gascony, are typically included in the popular Celtic nations branding the way Ireland, Scotland, and Wales are, despite carrying comparably deep genetic connections to the very same lineage running underneath all of them. Brittany carries its own version of this story next, with its own separate migration history layered directly on top. Brittany, tucked into the northwest corner of France, sits within the broader Atlantic modal haplotype zone, sharing deep R1b lineage roots with both Ireland and the Basque Country. Cornwall and Wales tell a similar story from the British side. Both regions were part of the original Beaker-affected British population, and both inherited the same near-total R1b turnover documented in the Olalde 2018 study, the same wholesale genetic replacement that swept across the rest of Britain roughly 4,400 years ago. Scotland adds its own data point to this pattern. Localized regional samples there show Rh-negative frequencies as high as 30%, matching Northern Ireland's own figures almost exactly, and reinforcing the Atlantic fringe pattern well beyond Ireland alone. All 4 of these regions, Brittany, Cornwall, Wales, and Scotland, are commonly grouped together under the modern Celtic nations label. The genetic data tells a different timeline. Their shared ancestry predates any historical Celtic identity by well over 1,000 years. Genetically speaking, the actual defining event connecting these regions has nothing to do with language or culture at all. It's the Bronze Age beaker-driven population replacement, occurring roughly 4,000 to 4,400 years ago, long before anyone spoke a Celtic language on any of these coastlines. The modern cultural Celtic grouping and the ancient genetic Atlantic grouping overlap heavily on a map. They were built by two completely different processes, separated from each other by thousands of years. That's the quiet irony sitting underneath most Celtic ancestry marketing circulating today. The genetics are real, measurable, and well documented. The label attached to them arrived over 1,000 years after the biology itself was already in place. Two more outliers on this map never get grouped with Ireland at all, and one of them isn't even in Europe. Corsica, Sardinia, and Crete were separately identified by early 20th century serologists as carrying unusually high ABO typo frequencies, the same marker that first tied Basque and Irish populations together in the earliest speculation about shared ancestry. These three Mediterranean populations sit geographically distant from both Ireland and the Basque Country, and there's no documented Bronze Age R1b replacement event connecting them the way there is across the Atlantic fringe regions. Their elevated O-type frequency is a genuinely separate case, a reminder that a shared blood type statistic doesn't automatically imply a shared migration history behind it. The Canary Islands present an entirely different case. The Guanches, the island's historical indigenous population, are now considered extinct as a distinct group. DNA testing of their modern remnant descendants points toward Basque and Berber ancestry instead. The same RHD deletion, Rh negative distribution model that first highlighted the Basque and Berber populations noted a direct genetic link between them. The identical complete RHD gene deletion appearing in both European and Berber populations. North African Berber populations of the Rif and Kabylia regions carry Rh negative frequencies of roughly 15 to 20%, the second highest cluster identified anywhere on Earth, after the Basque Peak. Put the full map together, and the real pattern stops looking like Ireland and one hidden twin. It becomes a documented, if uneven, Atlantic and Western Mediterranean genetic corridor with several real nodes scattered across it, of which Ireland and the Basque Country simply happen to be the tightest matched pair. Inside Ireland itself, that same corridor logic keeps repeating, at a much smaller scale. The 2017 Irish DNA Atlas project, led by Archia's Gilbert and colleagues, sampled somewhere between 194 and 196 Irish individuals, each one carrying 4 documented generations of ancestry tied to a specific region of Ireland. Combined with 2,039 additional samples drawn from the Peoples of the British Isles dataset, the analysis identified 10 distinct, geographically stratified genetic clusters within Ireland alone, a level of internal structure most people never associate with a single small island. 7 of these clusters represent what researchers classify as Gaelic Irish ancestry. 3 represent shared Irish-British ancestry, concentrated largely near historical points of British settlement. The study also identified a major genetic barrier running across the north of Ireland, in Ulster, a measurable divide showing up directly in the DNA, not just in the region's political or cultural history. Using a reference panel of 6,760 European individuals, alongside the 2 Cassidy ancient genomes from earlier research, the team detected high levels of both Northwest French-like and West Norwegian-like ancestry embedded within the modern Irish population. The West Norwegian-like signature is interpreted as genetic evidence of Norse Viking gene flow into Ireland, the first such confirmed evidence recorded at this level of resolution. Lead author Edmund Gilbert described the finding as showing Ireland functioning as a sink of Celtic ancestry, with genetic structure closely mirroring the historical medieval kingdoms of Ulster, Leinster, Munster, and Connacht, boundaries drawn centuries ago that still leave a visible trace inside the modern genome. That same research team's earlier ancient DNA work had already uncovered something far stranger, buried underneath one specific Irish monument. In 2020, Cassidy and colleagues published a whole genome analysis of 44 Mesolithic and Neolithic Irish individuals in Nature, including remains recovered from the Passage tomb at Newgrange. One individual, labeled NG10, showed genomic evidence that his 2 inherited genome copies were extremely similar, a hallmark of close inbreeding, later confirmed to indicate that his parents were first-degree relatives. Researchers initially interpreted this as evidence of a dynastic elite practicing socially sanctioned incest, comparable to Egyptian pharaohs or Inca god kings, as a method of preserving a bloodline's exclusivity. The same dataset uncovered distant familial links between NG10 and individuals buried at the Carrowmore and Carrowkeel Passage Tomb cemeteries in Sligo and at Millen Bay in County Down, sites separated by hundreds of miles. The same study also documented the world's earliest known case of Down syndrome, in a child buried around 3629 to 3371 BC at Pulnabrone, Ireland's oldest known burial monument. Not a single confirmed royal lineage emerged from any of it. What emerged instead was a far messier, more contested set of conclusions than the original 2020 headlines suggested. A 2025 follow-up study directly challenged the dynastic elite interpretation of the Newgrange find, arguing the evidence doesn't actually support a royal incestuous hierarchy the way the original coverage implied, an open scientific reversal that's still being actively debated. Whichever interpretation eventually wins, the underlying question is the same one running through this entire island's genetic record. Why does isolation leave such a clear signature at all? Ireland's genetic distinctiveness is repeatedly tied, in the literature, to one structural fact. Britain retained a land bridge to continental Europe after the last ice age, while Ireland was cut off by sea. That sea separation forced early Irish populations to arrive by primitive boat, in small founding groups. A classic setup for a founder effect, where a limited starting gene pool ends up shaping everything that follows for thousands of years. Reduced Irish haplotype diversity, elevated runs of homozygosity, and elevated linkage disequilibrium are all measurable modern consequences of that small, semi-isolated founding population, still detectable in genomes sequenced today. The Basque Country shares a comparable geographic logic of its own, a mountainous, historically hard-to-penetrate region at the edge of Iberia, which likely limited outside gene flow in remarkably similar ways. Two isolated populations, thousands of miles apart by land, arrived at strikingly similar genetic profiles, largely because they were both shaped by the same Bronze Age migration wave, hitting two separated, geographically defensible refuges at roughly the same moment in history. The 2021 study's own framing treats the Ireland-Basque-Gascony cluster as consistent with a real historical replacement event, not as an unexplained statistical coincidence sitting on a graph. Ancient DNA sample sizes remain small by modern standards. A few dozen genomes represent thousands of years of Irish history, and each new burial site sequenced has, so far, revised the story rather than confirmed it outright. So the real question isn't whether Ireland's blood matches the Basque Country. The data already answers that. The real question is how many more of these matches are still buried, waiting on the next genome.